Hello everyone, welcome. I'm Dr. Mythri Shankar, Associate Professor in Nephrology at Institute of Nephrology, Bengaluru, India. This talk is a practical update on the genetics and genetic testing of ADPKD for nephrologists. Here is a roadmap for the talk. We'll build from the foundations and the two core genes through the polycystic biology and how cysts form into why disease severity varies so much within and between families. Then the wider gene spectrum behind the atypical disease, followed by the practical part, that is when and how to test, choosing a genetic panel and interpreting a VUS and the risk scores also, and finally the most important, reproductive counseling. Some key terminologies which we should understand before delving deep into genetics of ADPKD. A disorder caused by variation in a single gene is called as a monogenic disorder. The fixed position of a gene on a chromosome is called locus. Two variants on the same copy is cis, on the opposite copies is trans. Both gene copies, maternal and paternal, carrying a variant is called biallelic. A variant that reduces but doesn't abolish the gene function is a hypomorphic variant. Truncating mutation cuts the protein short, for example, nonsense mutation or frameshift mutation. Non-truncating mutation alters the protein residue, for example, the missense mutation. For reasons, genetics is now a clinical topic and not just an academic topic. First, ADPKD is monogenic and mostly explained by two genes, so a single test is often informative. Second, genotype predicts the course, it has prognostic value. Third, testing has become actionable for reproductive planning, living donor evaluation, and atypical presentations. Fourth, the polycystin cyclic AMP biology is what tolvaptan and newer agents target, so the mechanism links directly to therapy. The KDIGO 2025 guidelines updates how we name the disease. The label now incorporates the gene where it is known, starting from an ADPKD phenotype with or without family history. Genetic testing sorts patients into groups. So if testing isn't done, if it is inconclusive or is negative, we still call it ADPKD. If a pathogenic variant in PKD1 or PKD2 or a confirmed minor gene is found, we would add the gene name as ADPKD dash the name of the gene. So moving forward, expect gene appended nomenclatures on the reports. PKD1 and PKD2 together account for over 90% of diagnosed families. PKD1 on chromosome 16 is roughly 78% of cases and encodes polycystin 1. PKD2 on chromosome 4 is about 15% and encodes polycystin 2, which is a calcium channel. A further 7% comes from rarer spectrum genes, which we will cover shortly. If you look at the population prevalence figure, it is around 1 in 1000, and this is based on the protein truncating variants. But it would be definitely higher if you count non-truncating likely pathogenic variants as well. PKD1 disease is more severe, forms earlier cyst, has faster kidney enlargement, and a median age at kidney failure is around 55. PKD2 is milder with failure closer to 70. Within PKD1, truncating mutations are worse than non-truncating ones. That distinction feeds directly into the pro-PKD score. So when a report comes back, note both the type of gene and the mutation type. A young patient with a PKD1 truncating variant deserves closer surveillance than a PKD2 patient of the same age. PKD1 testing has been historically difficult and also VUS are common when you receive the report. Why is it? That is because PKD1 is a large gene. It has 46 exons, a 13 kilobase coding sequence. Crucially, exons 1 to 33 are duplicated about 6 times as pseudogenes on chromosome 16 with very high homology. So you need locus-specific amplification to avoid contamination from those copies. High GC content also compounds the problem, and most pathogenic variants are private, that is over 1650 pathogenic variants are described, out of which many are ultra rare, which is why you will frequently see variants of uncertain significance. Long read sequencing, whole genome sequencing, and modern NGS have largely solved this technical hurdle. This figure maps the described pathogenic variants along PKD1 and PKHD1 proteins. If you look at PKD1, there is a sheer scatter and diversity of variants here, if you can see here. You find all kinds, that is missense, nonsense, frameshift, splicing, in-frame, insertion deletion distributed across a very large 4,300 amino acid protein with many functional domains. There is no single hotspot you can simply screen. Variants are spread across the whole gene and most families carry their own private mutation. This is exactly why comprehensive sequencing is needed and why variants of uncertain significance are so common in PKD1. In contrast, if you look at the PKHT1 below, it is a recessive gene with limited number and variety of disease-causing mutations. Now let's look at the mechanism. Polycystin 1 and polycystin 2 form a complex in the primary cilium, which is the little antenna on the tubular cells, where they regulate the intracellular calcium. When polycystin function is lost, cells switch to proliferative fluid secreting phenotype. Cyclic AMP is a central driver. It pushes both proliferation and fluid secretion into the cyst lumen. This is exactly why tolvaptan works, because it blocks the vasopressin V2 receptor and lowers the cyclic AMP. So the biology on the left side is the rationale for the therapy on the right. So common question from the students usually is that if every cell carries the mutation, why do only some tubules form cysts? This is because of the two-hit model. The inherited germline mutation is the first hit, which is present in every cell. A cyst forms only when the second or normal copy is also knocked out somatically in an individual tubule cell. That cell then expands slowly into a cyst, which explains the focal scattered pattern. Third hit is the environmental trigger and gene dosage effects. These account for unusually early or aggressive disease, including rare in utero presentations. Autosomal dominant inheritance, that is, each child of an affected parent has a 50% chance of inheriting the variant, and males and females are affected equally. About 10% of the cases arise de novo with no family history, so a negative family history does not exclude the diagnosis. Both males and females affected with ADPKD should receive genetic counseling. This CJASN study illustrates the wide variability in how PKD1 disease progresses between individuals. Even with the same gene involved, the rate of kidney enlargement and decline in function differ substantially. The point to make is that gene identity alone does not fully determine the outcome. The type of mutation, the dosage, modifiers, and environment, all of them contribute. These age-matched MR images show us the varying severity of genotype. All the top and middle panels are PKD1 truncating mutations at ages 36 to 49, showing progressively higher Mayo imaging classes and dramatically higher height-adjusted total kidney volume. Compare that with PKD2 patients of similar age who sit at much lower kidney volumes and milder imaging classes. So for the same age, a PKD1 truncating mutation produces markedly more cystic burden than PKD2. This is a genotype-phenotype correlation you see on the scan. This study published in KI report summarizes a familial variability in ADPKD severity. 92 families and nearly 300 affected individuals from the Irish Kidney Gene Project were studied. Marked intrafamilial variability was seen in about 13% of the families, meaning relatives sharing the same variant can still have quite different courses. Families carrying PKD1 non-truncating variants were overrepresented among those with variability. The average intrafamilial difference in age at kidney failure was around 7 years, and allelic type alone did not explain the onset. So the clinical lesson here is that genotype sets the baseline risk, but you cannot predict an individual's exact course from the family history alone. So then severity isn't determined by the gene alone. So what drives the variability in severity? First one is, as we already discussed, the type of mutation, PKD1 truncating at the severe end, PKD2 at the mild end. Allelic dosage matters, that is biallelic or hypomorphic combinations can produce very early severe disease. Mosaicism, where only a fraction of the cells carry the variant, tends to give milder or asymmetric disease and can complicate testing. And modifiers like genetic background, for example, polygenic or digenic inheritance pattern, plus modifiable factors like blood pressure, water intake, smoking, obesity, diabetes, uncontrolled hypertension, all of this will shift the pace. The last point is empowering for patients, because some of what drives progression is actionable and can be managed by the patients themselves. Beyond the two main genes is a growing spectrum we should recognize because the patients often look atypical. Glucosidase 2 alpha subunit, that is GANAB, GANAB, overlaps ADPKD and polycystic liver disease with mild kidney involvement. DNA JB11 gives smaller kidneys and a fibrosis overlap with late kidney failure. ALG9 and IFT140 cause comparatively mild cystic disease. IFT140 in particular can preserve function with atypical imaging. This table from CJASN lays out clinical scenarios of atypical ADPKD and the genetic explanations that may underlie them. These are typically the conditions where we would require genetic testing. For example, markedly asymmetric or unilateral disease, unexpectedly mild or severe course, very early or antenatal presentation, or a prominent liver phenotype. Each atypical pattern points towards a specific mechanism, that is mosaicism, a minor spectrum gene, digenic or hypomorphic combinations, or biallelic inheritance. This is a nice compact high yield summary figure from Kidney News. The first figure lists indications for genetic testing, both the common ones, that is absence of family history, early onset or atypical presentation, potential young kidney donor, pre-implantation diagnosis, and expanded ones, that is intrafamilial variability, imaging clinical discordance, low cyst burden with low GFR, extrarenal features suggesting a different syndromic cysticis. Figure B is a broader differential of genes that can mimic ADPKD. That includes nephronophthisis genes, ADTKD genes, ADPLD, developmental disorders, type 4 collagen-related nephropathies, syndromic or tumor-related genes. This figure ties the 8 ADPKD-related genes to genotype-phenotype correlations with the prevalence pie chart. It is a good reference for whom all to test and what the genes mean. The article from Kidney News by Dal NK et al. So you can see here that 52% is PKD1 truncating, 26% is PKD1 non-truncating, 15% PKD2, and the remaining 3% are all the minor genes listed here. This Kidney International table is a definitive gene-by-gene reference for the ADPKD spectrum. It gives for each gene the proportion of screened families, the disease designation under the new nomenclature, and the kidney and extrarenal phenotypes. Note PKD1 is split into truncating versus non-truncating rows because that distinction carries prognostic weight. And the minor genes, that is ALG9, DNAJB11, GANAP, IFT140, NEK8, each with a characteristic milder or atypical patterns, and comments on biallelic or mosaic associations. So this is a good resource to consult when a report names one of these less familiar genes. The continuation of the same, other disorders with present with kidney cysts and the genes and the mutations involved. So this was the first study to establish IF40 as a monoallelic cause of ADPKD. Whole exome and targeted sequencing across large PKD cohorts, including the 100,000 Genomes Project and UK Biobank. The investigators identified families with monoallelic pathogenic IFT140 variants, accounting for a good number of previously unsolved or sequencing naive families. The pedigrees and imaging on the left show the characteristic atypical, comparatively mild PKD phenotype. IFT140 was validated as effectively the third most common PKD locus. This study by Senam et al. Compares kidney function and kidney size between IFT140 and PKD2 mutations. IFT140 patients usually retain relatively preserved renal function, yet they can have enlarged kidneys with an atypical imaging pattern, that is Mayo class 2A rather than the typical 1 series. So IFT140 disease can look structurally significant on imaging while function holds up better than you would expect. This is a useful pattern to recognize so you don't overcall the prognosis and label it as poor. GANAP was identified through whole exome and Sanger sequencing in a set of families. The gene encodes the alpha subunit of glucosidase 2 and the defect impairs polycystin 1 maturation. The phenotype is characteristically mild on the kidney side, an average of only around 10 cysts with patients typically in CKD stage 1 to 2, while polycystic liver disease can range from mild to severe. GANAP should be considered when kidney involvement is mild but liver cysts are prominent. DNA JB11 was established as cause of ADPKD by Cornick Legal and colleagues. It has smaller rather than massively enlarged kidneys with a fibrocystic and interstitial component. DNAJB11 encodes a chaperone protein and its dysfunction is associated with late onset kidney failure. Clinically, this matters because DNAJB11 patients often don't show the large polycystic kidneys of typical ADPKD. Kidneys can be normal sized or small with fibrosis, yet still progress to end-stage kidney disease at older ages. Keep DNAJB11 in mind for a patient with cystic and fibrotic picture and late kidney failure who don't fit the usual ADPKD imaging. The JASN visual abstract characterizes ALG8 and ALG9 as cystic kidney and liver genes with limited penetrance across more than 3,900 PKD and PLD families plus population biobanks. Monoallelic ALG8 and ALG9 pathogenic variants were enriched several fold over non-PKD controls. Most carriers had kidney cysts around 89 to 90%, but usually a low cyst burden. And importantly, their eGFR did not differ significantly from controls. So these are lower penetrance genes for significant functional decline, and finding one explains this but generally predicts a milder course. An important counseling point is that sporadic cases without any family history occur in roughly 10 to 20% of the families. Much of this reflects the de novo variants, that is a mutation arising in a patient rather than inherited from a parent. The practical implication is that a negative family history does not exclude ADPKD. De novo case still carries the standard 50% transmission risk to that patient's own children. Mosaicism is the other reason a case can look sporadic or atypical. If the PKD1 mutation arises after fertilization, only a fraction of the patient cell carry it. The diagram shows gonadal, low and high grade somatic and mixed patterns. Clinically, mosaicism tends to give milder or asymmetric disease and can be missed by standard sequencing because the variant is present at low allele fraction, so it may need deeper next generation sequencing to detect. Gonadal mosaicism is passed on to the future generations, while somatic mosaicism is not. Recognize mosaicism when disease is unexpectedly mild, asymmetric, or apparently de novo. This is the conceptual heart of the dosage model. Cystic disease severity tracks inversely with the total functional polycystin dosage running from normal through very mild, moderate, severe and lethal. A single heterozygous PKD1 or PKD2 variant sits towards the milder end. Combinations that further reduce polycystin, that is a hypomorphic allele in trans, a digenic PKD1, PKD2 involvement or biallelic variants push severity progressively higher with most severe, potentially lethal disease at near complete loss. This explains very early or antenatal presentation and reframes ADPKD severity as a continuum of residual polycystin function. How to actually deliver genetic testing well? This workflow runs from patient identification, identifying which patients you want to test, performing a pre-test counseling and discussion of value of genetic testing. Following that, genetic test is ordered, you should know which test to order. Then once the results are obtained, in-house evaluation of genetics in the nephrology genomics board is important, which involves nephrologists, pathologists, molecular geneticists, genetic counselors, research coordinators, clinical geneticists and program manager. Then the report of the results is returned to the patient with post-test counseling and referring cascade testing is also considered in the family members. You can also save the patient's blood with the permission of the patient for research, follow-up, broader genetic analysis or omics studies. How to approach testing in practice? Imaging with age-adjusted Pi-Ravine criteria still diagnoses most patients without any genetics. Reserve molecular testing for defined situations, that is atypical or uncertain presentations, a young at-risk person being considered as a living donor, reproductive planning, or when you specifically need prognostic information. Method-wise, an NGS panel covering PKD1, PKD2 and the spectrum genes is the first line with exome or long-read sequencing when the panel is uninformative and suspicion remains high. Then report, counsel and restratify. Before you order, know exactly what the panel covers. This is frequent avoidable pitfall. As an example, my personal preference is Okay, if you look at some of the examples, Met genome PKD gene panel does not include IFT 140, while whole exome sequencing covers all major and minor genes. Metropolis PKD gene panel covers the following genes. I personally prefer whole exome sequencing over a limited panel, precisely so you don't miss genes like IFT 140. Always read the gene list before ordering and match it with the differential diagnosis. Some tips that can increase the yield. First, always order the test with VUS reporting included, because so many PKD1 variants are ultra rare and private, and genuinely causative variant may not meet the normal formal threshold for pathogenic or likely pathogenic and would be filtered out if VUS are suppressed. Second, have access to an expert, a genetics colleague or genomics board for when results are ambiguous, because VUS interpretation and segregation analysis are where nephrologists most often get stuck. This is an example of our patient. The patient presented with advanced disease, hypertension, EGFR under 15, markedly raised creatinine and urea, large kidneys, and a family history of kidney disease. Clinically, a strong ADPKD picture, yet the report returned only a variant of uncertain significance, a PKD1 missense change. This is the real world scenario. The VUS algorithm was built for this, that is high pretest probability plus a VUS. This is the anatomy of genetic result. The first section shows the affected gene and the exact reference transcripts, that is the ensemble and the refseq, the coordinates that are referred to. Second is the location where the gene, the change in the gene sits. Here it is exon 37. And then we have the variant. C dot is the DNA level change and P dot is the protein level change. Here we have a missense substitution. Zygosity, heterozygous is one of the two gene copies is affected, consistent with dominant disease. And then we have the OMEM number, the linked condition and its unique OMEM catalog number. We have the inheritance, how the condition is transmitted, which is autosomal dominant. Then we have the classification, that is ACMG call, which is uncertain significance. VUS, the code list evidence is also applied. So these are the evidence codes. VS is very strong, S is strong, and so on. This is a practical algorithm for interpreting a VUS in a patient with suspected ADPKD but no pathogenic or likely pathogenic PKD1 or PKD2 variant. Start by assessing the pretest probability from a clinical, renal, and extra-renal point of view. If it's not high, then stop. If it is high, first ask whether another cystic kidney gene explains the phenotype. If so, stop and pursue that. If not, look for a VUS in PKD1 and PKD2, then check its rarity. A minor allele frequency below 1 in 10,000 across all subpopulations is supportive. If it's commoner than that, it's unlikely to cause significant disease, so stop. If it is rare, check whether it is reported in the Mayo PKD database. If yes, use that to predict clinical significance. If no, escalate to in silico prediction tools and screen additional family members for segregation. So based on this, we looked at the population database, that is the NOMAD population database, and we found that it was extremely low in frequency. The mutation was non-truncating, non-synonymous variant located in a mutational hotspot, which was critical and well-established functional domain. A reputable source had recently reported the variant as pathogenic. So reclassification showed that the variant was likely pathogenic. In India, genetic testing for suspected ADPKD is usually not covered by most health insurance. A standalone diagnostic and genetic tests done for evaluation or screening are commonly excluded, though the IRDAI rules do prevent insurers from refusing a policy to someone because they carry or at risk of a genetic disorder. That's about the access to insurance, but not reimbursement of the test. Testing may occasionally be reimbursed if the patient is hospitalized, that is in-hospital billing, and deemed medically necessary for treatment. Advise patients to check their policy's exclusions for genetic testing and investigations, or to ask the insurer in writing. Given near complete penetrance, reproductive counseling should be proactive and offered to both men and women. There are few options available. Number one, natural conception with awareness of 50% transmission risk. Second, prenatal diagnosis by chorionic villus sampling or amniocentesis. Third, pre-implantation genetic testing, that is IVF with PGT-M to select the unaffected embryo. Cost is a real barrier here. Then we have donor gametes and adoption are also on the table. And finally, the decision to go childless. Counseling should be non-directive. Your role is only to lay out options clearly and not to steer the decision. To summarize the 5 things to carry into clinic: 1. PKD1 and PKD2 explain the large majority, but keep the spectrum genes in mind for atypical cases. 2. Genotype predicts severity. PKD1 truncating has worst, PKD2 mildest, and feeds Mayo and pro-PKD. Third, test with a clear purpose rather than reflexively. 4. Interpret a VUS using pretest probability, family segregation, and rarity, and revisit it over time. 5. Raise reproductive options much earlier in the disease duration. These are some of the key references. Thank you for your patient listening. This is my email ID and my Twitter account in case you want to reach out for any doubts.